my name is Anna Cisielska. I'm a postdoc in the Laboratory of Molecular Membrane Biology at the Nęcki Institute of Experimental Biology in Warsaw. Studies conducted in our laboratory are focused on molecular mechanism of activation of plasma membrane receptors involved in immune cell functioning. We pay special attention to toll-like receptor 4, which triggers pro-inflammatory signaling pathways. TLR4 is activated by lipopolysaccharide, a component of gram-negative bacteria. For that reason, TLR4 is involved in combating bacterial infection. An exaggerated immune response to LPS can cause potentially fatal systemic inflammatory condition named sepsis. Moreover, TLR4 activity has been linked with chronic inflammatory diseases such as atherosclerosis. TLR4 is a plasma membrane receptor and its activity is determined by the organization of the membrane. The plasma membrane is a bilayer composed of proteins and lipids. The lipid composition of the plasma membrane can be modulated by lipids from the extracellular milieu. In the human body, such membrane-modifying lipids and lipophilic compounds are obtained from food or are produced in situ under some pathological conditions. In our review published in BioEssays, we discussed how dietary components can affect plasma membrane organization that leads to changes in the signaling of TLR4. Dietary components can have either positive or negative influence on human health. The distinct domains of the plasma membrane enriched with cholesterol and sphingolipids, named rafts, play a critical role in TLR4 signaling. Rafts are nanoscaled, highly dynamic structures, and from physicochemical point of view, they are more ordered and less fluid than the surrounding membrane enriched in glycerophospholipids. There is a distinct set of membrane proteins associated with raft domains. One such protein is CD14, highly abundant on the surface of macrophages. CD14 binds lipopolysaccharide, LPS, and it is involved in TLR4 activation. In contrast to CD14 in unstimulated cells, TLR4 does not associate with raft domains. During bacterial infection, LPS aggregates, which are present in blood serum, are recognized by LPS-binding protein, LBP. LBP facilitates extraction of LPS monomers by CD14. Subsequently, CD14 shifts the LPS to MD2 protein associated with TLR4. Many data indicate that TLR4 associates with rafts upon LPS binding. In the next step, TLR4 undergoes internalization. Recruitment of different adaptor proteins by TLR4 triggers signaling cascades leading to production of cytokines and inflammatory responses. Taking into account the dynamics of activated TLR4, the plasma membrane, it becomes clear that the organization and fluidity of the membrane have strong impact on TLR4 function. As it has been said, properties of the plasma membrane can be changed by dietary components. This group of compounds includes cholesterol and its oxidized derivatives, fatty acids, α-tocopherol, and plant polyphenols. By changing dietary habits, we can either stimulate or suppress inflammatory reaction of our immune system. The typical westernized diet is rich in sugars, cholesterol, and saturated fatty acids. High amount of the two latter are found in foods from animal sources. Ample data indicate that a high cellular level of cholesterol and saturated fatty acids upregulates LPS-induced pro-inflammatory signaling of macrophages by facilitating assembly of lipid rafts. Recently, epidemiological studies have indicated that pro-inflammatory properties have manufactured trans fatty acids which are present in processed food. Immunostimulatory properties possess also oxidized derivatives of cholesterol, so-called oxysterols, which can be obtained with the diet, but mainly are produced by oxidation of LDL inside of oxidative stress, such as atherosclerotic lesions. Oxysterols, like 7-ketocholesterol, can substitute cholesterol and form nanodomains with sphingolipids. The lipid domains formed in the presence of the oxysterol could promote association of TLR4 with CD14 and or its dimerization in the presence or even in the absence of LPS. A therapeutic effect can be achieved by enrichment of the diet with compounds that restore the balance in the content of the plasma membrane rafts and thereby increase membrane fluidity, ultimately having anti-inflammatory properties. The most promising in these regards are N-free polyunsaturated fatty acids, α-tocopherol, and plant polyphenols. Recommended sources of N3 polyunsaturated fatty acids are seafoods such as fish and shrimps. The biological activity of N3 polyunsaturated fatty acids was proposed to result from their incorporation and disassemble of the rafts. As a result of raft disorganization, the association of TLR4 with CD14 can be abolished, and this can suppress inflammatory responses. You are what you eat. This well-known phrase properly describes the phenomenon of diet-related effect on acute and chronic inflammation. And it works. An excellent example is our cat. Supplementation of her diet with PUFA and tocopherol, of course ordered by the vet, improve her fur and even her mood.